So micro and nanoplastics are everywhere in the food we eat, the water we drink, and the air we breathe. When they enter the bloodstream, these particles can circulate through the body. They've been detected across multiple tissues and organ systems, including the liver, kidneys, heart and blood vessels, placenta, and breast milk, as summarized in a recent review published in Nature Medicine. These particles can also accumulate in the gastrointestinal tract, lungs, and endocrine and reproductive organs, where such accumulation is associated with potential disruption of the normal biological function. As someone with over 2 decades of experience in chemical engineering, I find this evidence deeply concerning. Micro and nanoplastics enter the human body continuously through ingestion, thermal contact, and inhalation. Exposure to these particles occurs without interruption. Now, among these pathways, inhalation may represent a particularly hazardous route. According to a study conducted in a coastal city in eastern China, individuals spending time outdoors may inhale up to 1 million microplastic particles per year. Recent findings suggest that when inhaled, the plastic particles, particularly those carrying surface charge, may migrate along the olfactory nerve through the brain toward the brain where measurable concentration have been detected. Among the organs examined in a recent study, the highest concentration of micro nanoplastics were detected in the brain tissue. The levels there were 7 to 30 times higher than those in the livers or kidneys. The researchers at the University of New Mexico estimate that in middle-aged individuals, each gram of brain tissue contains about 4,900 micrograms of plastics, roughly 0.5% by weight. In absolute terms, this amounts to about 7 grams, comparable to the weight of a small plastic spoon. Here, I want to point out that the situation may be even more concerning for children because their lower body mass and higher intake of air, liquid, and food relative to their weight can result in substantially higher exposure to plastic particles per kilogram, up to 4 times greater than adults by some estimates. This brings us to question whether internal accumulation of these particles is associated with measurable effect on human health. According to the European Academy of Neurology, residents of coastal areas with higher levels of micro nanoplastics showed increased prevalence of functional impairments, including 9% higher cognitive disability, 6% higher mobility disability, 16% higher self-care disability, and 8% higher independent living disability. While these findings do not establish causation, the associations are consistent with emerging mechanistic evidence and indicate that these particles can reach the brain, induce neuroinflammation, and disrupt cellular homeostasis. But the potential impact of micro nanoplastics is not limited to the particles themselves. Beyond their physical presence, micro nanoplastics may pose additional risks because they also carry chemical additives, including phthalates, bisphenols, polybrominated diphenyl ethers. Health impacts associated with just 3 of these additives were estimated at $1.5 trillion in 2015. Note that more than 16,000 chemical compounds are used in plastic production, many of which have not been comprehensively evaluated for long-term health human effects. Beyond chemical additives, another factor that may contribute to the biological impact of micro nanoplastics is surface charge. Plastic particles acquire and retain surface charge through the friction in air, also known as triboelectric effect, or by absorbing ions in aqueous environments, including biological fluids. The surface charge enhances interactions between these particles and cells, organelles, and biomolecules such as DNA and proteins. The smaller the particle, the stronger the surface-driven interactions, potentially amplifying their biological impact. As noted in a recent peer-reviewed article, plastic particles may pose significant environmental and health hazards, with surface charge playing a critical role in governing their behavior. An important consequence of these surface interactions emerges when the particle enters biological fluids. As described in a growing body of research, micro nanoplastics are rapidly coated by proteins, lipids, and other biomolecules, forming what is known as eco-corona. As shown here, the biomolecules gather around the particles in the bloodstream, forming this corona. This molecular coating can alter how the particles are recognized by cells and the immune system. Rather than being identified solely as foreign materials, they acquire a new biological identity determined by the molecule bounded to their surface. As a result, these particles may be internalized more readily by the cells. Studies suggest that micronanoplastics, particularly smaller nanoplastics, can cross several of the body's key protective barriers, including the intestinal barrier, the pulmonary barrier, the blood-brain barrier, and the placental barrier. Once these are across these barriers and within the tissues, these particles are not readily cleared and they may persist for prolonged periods. In addition to persistence in the body, micronanoplastics may also increase viral stability. Viruses exhibit strong adsorption to plastic particles, and nearly 99% of the viral dose in some experiments was bound to their surfaces. Those that normally would lose viability rapidly may remain detectable on plastic surfaces for several days, and up to a week in some cases. Through this surface binding, micronanoplastics may function as virus carriers, potentially contributing to prolonged viral persistence and influencing transmission dynamics. Here, it is important to note that these particles also exert direct biological effects. Research findings indicate that charged micronanoplastics may disrupt tightly regulated cellular processes essential for normal cell function and survival. They may affect intracellular signaling, activate pathways involved in programmed cell death, and impair mitochondrial function, thereby affecting cellular energy production.